All right, and welcome again to Biology 2. This is the start of our third quarter. We'll be discussing two important topics for the rest of this quarter. We'll be starting first with proteins, and then after that, we'll be continuing our discussion on nucleic acids. Let's begin with the first module. With the first module, we have this question: which came first? Is it the chicken or the egg? So probably you have heard about this discussion or about this debate, and there will be people who will be saying it's the egg, and there will be people who will be saying it is the chicken. But for recently, there has been a research or paper that was published, and it says that scientists finally concluded that the chicken came first, not the egg, because the protein which makes the eggshell is only produced by a hen. And this protein is called ovoclidin 17-17 or OC17, which catalyzes the development of eggshells, can be only formed in the ovaries of hens. So this would be those who would be accepting that the chicken came first. While on the other hand, there will be people on the other side of the camp, particularly this was posted by Neil deGrasse Tyson, and he said through a message that just to settle it once and for all, which came first, the chicken or the egg? The egg laid by a bird that was not a chicken. And this is more on the perspective of evolution, saying that the egg came first because it was laid by a proto chicken or by an organism that is not a chicken but later laid an egg that became the chicken later on. In any case, the debate is still ongoing, so we still don't know. We'll just be waiting for other evidences to come out. But in any case, the reason that we make use of this is because, as mentioned a while ago, proteins would be very, very important here. So with that, let's talk about the first topic of the third quarter, which would be proteins, and particularly in module 1.1, we'll be talking about amino acid monomers and polypeptides. And our objective for module 1.1 is number one, to describe what is a protein and identify its major functions. So that's why we need to take note, or you need to take note, all of the major functions of the proteins. Number two, determine the basic structure of amino acids. So you need to take note of the four important parts of the amino acid. You need to memorize this. And the third one is explain the importance of the variable R group, and you will be later learning what would be the importance of this variable R group that is part of the basic structure of the amino acid. Okay, so these are examples of proteins. You're very familiar with this. So you have the fish, you have meat, probably this pork meat, nuts, and chicken meat, and then several other sources of proteins. And then if you're going to be looking inside of the cell, you will have this protein, for example, this one that would be catalyzing or producing the RNA from the DNA, and that one would be what we call as a protein molecule. And there are other such examples like this plant-based protein sources, so it would be very, very good if you are familiar with this. So this would include beans, broccoli, chickpeas, grains, lentils, nut butter, nuts and seeds, peas, okay, potatoes, and several others of this like tofu and veggie patties. All of these are plant-based protein sources. So if you are a health nut, or you want to be, or you're very concerned with your health, plant-based proteins can be a substitute for animal-based protein to keep yourself healthy. Now, in case of the animal-based protein, you're very familiar with a number of this. You have the eggs, uh, chicken, turkey, prawns, tuna, salmon, pork chop, duck rib eye, and then Greek yogurt, cheese. This will be semi-skim milk. This will be all sources of protein that are animal-based. Now, the first objective that we have for this module is for us to learn about the functions of the protein. So there are 8 of them, major functions that will be discussed. So just remember this one and hope this will be a mnemonic that will be helpful for you to remember. Threads at sea, thread C, which is THRDEDSSC. So let's look at the functions of the proteins. What is letter T? T is transport. H is hormone. R is receptor. E, enzyme. D, defense. S is for storage, S is for structure, and finally C is for contractile and motor or contraction in another sense. So these are the main functions of the proteins. Threads C again. So that will be transport, hormone, receptor, enzyme, defense, storage, structure, and contraction, contractile, and motor. And this will be the things that you have to take note of this. So here are some examples of those proteins and their functions. So for example, the hormones, so you have your insulin over here, you have the protection, the antibodies will be very important part of the immune system that will take care of our body. So any kind of bacteria or virus that will come inside our body will be destroyed by the immune system. And then you have the transport, hemoglobin particularly, which transports oxygen. For enzyme, here is rubisco, that is very important. Storage, permethrin, spider silk structure, so you're very also familiar with the cobwebs produced by spiders, and this would be a protein as well. The receptor like rhodopsin, which is found in our eyes, very important for receiving light, and contractile, that is examples of this are proteins called actin and myosin, which are the two important proteins that makes up our muscles. So those are examples of the different functions of the protein. So we have, again, here's the enzyme over here. So we will be discussing that in module 1.3. Another example of storage molecule would be casein, which is a protein for milk. And then for eggs, it contains ovalbumin, which would be amino acids for the embryo. So these are important storage materials, proteins that will be storing not only energy but also amino acids. Then you're also familiar, again, as mentioned, antibodies for the fighting against diseases. We gave an example a while ago for transport. So in this case, this is a protein transport in the cell membrane, which causes some materials or atoms, elements to pass through from the outside inside to the inside of the cell, and then several others over here. So hormones like insulin is mentioned. Receptor like this would be involved in the signal pathway from the brain, from the different brain cells, from one brain cell to another one. So the dopamine can be produced here that will be received by the next brain cell that will proliferate the electrical signal of the brain. And then you have the contractile muscle, so particularly the actin and myosin, as I mentioned a while ago. This is what they look like. So this would be part of the muscle fiber and structure like collagen. That is another example for that. Okay, let's now go to the second important objective of our module, which would be the parts of the amino acid. There are four of these that are important. You can find this on the right side, the left side, the top side, and also the bottom side. Starting with the right side, what do you call this part of the amino acid? It is called carboxyl. While on the left side, you find here the amino group. On top, you will find here the R group or the side chain, the variable R group, and finally over here you have the H. Now take note that the carboxyl, the H, the hydrogen, and the amino acid never changes. It is only the variable R group or side chain, as mentioned with the word variable, that changes, and that actually makes amino acid different from each other. We'll be talking about that a little while. But with that, we have to take note of the classes of amino acids. So this is what we're talking about. What are the classes of amino acids that will be a result of the differences of the R groups among amino acids. So just remember NPAB, and the first one is what we call as nonpolar side chain. So the variable R group is a nonpolar, and then you have the, if you have a nonpolar, you have the polar side chain, which is hydrophilic, while the nonpolar is hydrophobic. We also have an A, which is the acidic, which is negatively charged, and letter B, if you have acidic, of course, you will have the basic, which is positively charged. So these are the four classes of amino acids that will be resulting with the variations that can be found among the R group. Let's look at some examples of this. Let's look at the nonpolar side chain. And the nonpolar side chain is said to be hydrophobic, which means they do not mix with water or any other polar substances. An example of this, glycine, alanine, valine, going down here, tryptophan, proline, and you will be noting something very important is that all of the variable groups, or most of the variable groups here, would contain carbon and hydrogen. So that will be what is very, very characteristic about the nonpolar side chain. Sulfur might be here, but it's in very middle, so it doesn't really affect the effect of this R chain. And here is an aromatic group here, over here, and there are several chains, rings that you can find here, but it is overwhelmingly made up of carbon and hydrogen, and that actually makes it non-polar and hydrophobic. On the other hand, we have polar side chains, which is hydrophilic, so this would be attracted or dissolving in water and other polar substances, and you will be noticing an OH, OH, an SH, an OH over here, an NH2, and NH2. So this would be making it hydrophilic. So this will be allowing it to mix with water. The acidic amino acid, on the other hand, it is negatively charged, and particularly you will be noticing the COO, which is the functional group called carboxyl, and that actually makes it acidic, related with acetic acid and all of those, which contain the COOH. And finally, you have the basic here, which is positively charged. So if the acidic is negatively charged because of the O minus here in the COOH, we have here the NH3 plus, NH2 plus, NH plus, and this would be making it positively charged and making that basic. So again, you have the N-pub, the non-polar, the polar, the uh, the acidic, and also the basic. So those are some examples of the different classes of amino acids. So we have now proceeded with the third objective of the module. We're finished with that because we now know what would be the fourth objective of the module, because we now know the differences or the effect of the differences with the variation of the side chain. So let's just finish up with this one, and then we have some concept checks a little bit later. The polypeptide formation. How does this form? Again, it is formed by a dehydration process, as we have mentioned in the previous module, wherein the polymers are actually made up of monomers that have reacted together with the release of water, and this is called the dehydration process. So here is one amino acid connected with amino acid, and the connection or the bond between them is called the peptide bond. And then if you're gonna look over here, here is the amino acid, and another amino acid needs to be connected with this, or the bond will be forming. There will be the release of water and the new peptide bond forming, and this is called the dehydration process. That's very important. So let's now look at some concept check. The 20 amino acids can also be classified as essential and non-essential. What does this mean? So what's the difference between the essential and non-essential? The difference would be the essential is also termed as indispensable, or meaning indispensable, and cannot be synthesized by our body, while the non-essential can be synthesized by the body. In other words, the essential amino acid, since it is not being produced by our body, we need to source it out from external sources or from our diet, so that if you do not have this amino acid, the essential amino acids in our diet, it's not found in the food that we eat, we're going to be having diseases, because we are not giving or we are not supplying the amino acids that are not being produced by our body. On the other hand, the non-essential amino acids are amino acids that even if they are not found in our diet, we don't eat this used through our food, it's okay. There will be no diseases, there will be no complications by the very fact that our body is actually producing this. So you really don't need to source it out from outside. Okay, another question. Which of the following is not a function of proteins? A. Transport molecule, acting as messenger, support the structure, store genetic information. The answer to this is letter D, because letter D is the function of nucleic acids. What monomers join to form polypeptides? Fatty acids, nucleotides, amino acids, or monosaccharides? And the answer to this is letter C, because amino acids form polypeptides. Another question, which of the following is not a basic component of every amino acid? So remember the four important parts of the amino acid. The answer is A, B, C, or D. The answer is letter D, hydroxyl group, because the R group, the amino, and the carboxyl group are all important parts of the amino acid. What chemical bond links the amino group of one amino acid to the carboxyl group of another? So the answer is it ionic, peptide, covalent, or hydrogen? The answer is it's letter B again. It's peptide bond. So we discussed this a little earlier. Another question, give the molecular formula of the functional group that is missing. So here is a picture of the amino acid. So the answer is A, B, C, or D. What is the missing part? The missing functional group is letter C. It is COOH. Another question, why are R groups important in amino acids? What do you think would be the answer to this one? Well, as we have discussed a while ago, it is the variety of the R groups that will be determining the properties of the amino acid, which are essential in the formation of the different chemical interactions with the polypeptide chain to achieve a certain level of conformation, making the protein functional according to its unique structure. So in other words, in the next module, we'll be discussing about the different level of structures of proteins. The functional groups will be very, very important because the interaction of this functional group will produce the higher levels of structure for proteins, and that would be also very important. And also, as mentioned, it is the functional group that will give the different properties of the amino acids, as mentioned a while ago, the NPAB, the non-polar, the polar, the basic, and also the acidic is also dependent on the R group as well. Okay, and that ends our discussion for this part. We'll see each other next time in